Good morning, everyone. Welcome to your channel, The Anatomy Canvas. Today, we are going to embark on a fascinating journey through the embryological development of the endocrine glands in the human body. So, as you know, in most textbooks, development of these glands is scattered across the multiple chapters, sometimes leaving us with the bits and pieces of puzzle, so rather than a complete picture. So I have gathered all these scattered pearls of the knowledge from the different parts of the books, and today we will weave them into a single continuous thread. So in just one session you will be able to understand the development of the endocrine gland in detail. So for the today's class, the learning objective are development of the pituitary gland, the thyroid gland, parathyroid gland, and the adrenal gland. Here you can say this is the timeline of the different glands development in the human body. So in different endocrine glands which are present in the human body, the testes they start development in the 7th week of the gestation, in the 7th week of embryonic life. Pancreatic islets starts development at 12th week. Adrenal glands start development at 6th week. Parathyroid gland development starts at 5th week. The thyroid gland development starts at 4 weeks. This is one of the first gland which develops. The pineal gland starts at 7 week, and pituitary gland starts development at the 4th week again. So out of all these glands, we have covered the gonads, testes, and ovaries in the lecture on reproductive embryology, and the pancreatic islets. I have covered this topic in that of the GIT embryology. So remaining glands we will cover it today in this class. First of all, the development of the pituitary gland, also called the hypophysis cerebri. So pituitary gland is an organ of dual origin. This is also an important MCQ you must know. The anterior part of the pituitary gland, it's developed from the embryonic buccal mucosa. This is called the adenohypophysis. So in pink color over here you can see the buccal mucosa, which its endothelium thickens, and then it forms an outpouching called the Rathke's pouch. So this Rathke's pouch give rise to anterior part or the adenohypophysis of the pituitary gland. Similarly, the posterior part of the pituitary gland, it develops as a downward growth from the diencephalon. This is called the neurohypophysis. So here in this picture you can see initial the thickening, then outpocketing. This outpocketing comes into contact with the downward growth of the diencephalon. Later on, this Rathke's pouch, its connection is lost from that of the buccal mucosa or oral mucosa. Here you can see they are in close opposition to each other, forming a bilobed gland, that is the pituitary, and here you can see the sphenoid sinus which is present over here. So what is the histogenesis of the pituitary gland? The pituitary gland begins during 4th week of development. This is again your MCQ. Thickening in the wall of the oral mucosa. Here you can see thickening basically starts at 3rd week, and after this thickening, when Rathke's pouch appear and it approaches the developing neurohypophysis, that is the 4th week of the development. So at the same time, downward extension of ventral diencephalon form posterior lobe, and two nascent lobes connect to form the bilobed structure of the adult pituitary. In this diagram you can see this one is the Rathke's pouch and this one is neurohypophysis. These two in enlarged view over here you can see this Rathke's pouch, also called the hypophyseal diverticulum of the oral mucosa. These two coming into contact with each other. Here you can see the connection of the hypophyseal stalk or the Rathke's pouch, which has been disappeared from the pharyngeal mucosa, and here you can see the initial bilobed structure of the pituitary gland. So Rutgers pouch constricts at its base and eventually separates altogether from oral epithelium during the gestational week 6 to 8. So this is the time, that is the 6 to 8 week, when it starts separating from the oral epithelium and this connection is lost. Another important point is this Rathke's pouch has the anterior wall and the posterior wall. So this anterior wall gives rise to the adenohypophysis or anterior lobe of the pituitary, while this posterior wall, while coming in contact with the neurohypophysis, this posterior wall will give rise to the intermediate lobe of the adenohypophysis, and the pars tuberalis, this over here is the, this area is the pars tuberalis. This pars tuberalis is an upward extension of the adenohypophysis that surrounds the lower region of the hypophyseal stalk. So the three parts, the anterior part forming hypophysis, posterior part of this Rathke's pouch forming intermediate lobe, and upward extension forming the pars tuberalis, which will later on surround the stalk of the pituitary gland. So what are the different congenital anomalies which can be related to the hypophyseal defects? This may be the pharyngeal hypophysis or cranial pharyngiomas. So pharyngeal hypophysis, you can see over here that this location, at this location you can see in the area of the pharynx, you can see the remnant of the first part of the Rathke's pouch, which can lead to the pharyngeal hypophysis. Similarly, craniopharyngiomas usually form near the region of the sella turcica or sometimes just above the hypophysis cerebri. So here you can see this craniopharyngiomas which are present. This is just above the pituitary gland, and this can lead to not only it can lead to the hydrocephalus, but also it can affect the development of the pituitary gland and leads to deficiencies in the levels of the hormones of the anterior pituitary, especially the growth hormone. Next is the thyroid gland. So thyroid gland is a small gland with significant defects. The mature thyroid gland is located in neck and is responsible for delivering hormones to the body. So important thing about the thyroid gland is that this is the first endocrine gland to develop in the human embryo. It begins to form about 24th day after fertilization, and it develops from the median endodermal thickening in the floor of the primordial pharynx. So here you can see in yellow color, you can see this is the primordial pharynx which is developing, and these are the pharyngeal arches 1, 2, 3, 4, and on inner side you can see the pharyngeal pouches which are present. So this gland, it develops from the primitive pharynx in the floor of the primitive pharynx. So this area is basically the location of the thyroid primordium. Thickening zone then forms a small outpouching. Here you can see a small outpouching as well, called the thyroid primordium. This diverticulum then descends inferiorly to reach its final destination in neck. Traditionally, the thyroid is located inferior to thyroid cartilage, approximately at the level of C5 to T10 vertebra, which is also your MCQ as well. So during its descent, the thyroid connects to the tongue by the thyroglossal duct. In this slide, you can see the thyroid originating from the between the first and the second pharyngeal pouches from the primitive pharynx, and it's forming the thyroid primordium. So in third week of gestation, around day 20 to 24, endodermal cells of the primitive pharynx proliferate, and they are forming the thyroid diverticulum. By beginning in the fifth week of the gestation, thyroid diverticulum migrate caudally. So it starts to migrate caudally in the fifth week of development. Along the midline, it crosses anteriorly the hyoid bone and the laryngeal cartilage to come to lie at its final position. So, first thyroid primordium is hollow, but it soon becomes solid and divide into right and left lobe in fifth week. This is again your MCQ as well. That it develops into two lobes at 5 week of development. These two lobes connected to each other by the isthmus. In this area, you can see this was the area between the first and the second pharyngeal pouches from where initially thyroid primordium have started. Then this thyroid primordium going downward anteriorly in front of the hyoid bone, and then the cartilages of the larynx to comes to lie its final position. So this connection is called the thyroglossal duct. Later on this connection is lost and it can be traced on the tongue at the position of the foramen cecum. The two lobes are formed in the fifth week of development, and these two lobes, they are connected to each other by the isthmus. Important point another is that isthmus lies anterior to developing second and third tracheal ring. This is also important, and by seventh week it assumes the definitive shape and has reached its final site in the neck. This is again MCQ as well. So also during the fifth week, the ultimobranchial bodies arise from the fourth and the fifth pharyngeal pouches. So here in this area in blue color, you can see the ultimobranchial bodies which are developing from the areas of the fourth and fifth pouches. These bodies ultimately differentiate into parafollicular cells, which plays an essential role in the calcium homeostasis. So the origin of the parafollicular cell is ultimobrachial body. This is another very important point, because you must know initially it was thought that these parafollicular C cells are derived from the neural crest cells. So the recent work have disputed this by suggesting that they arise from the endoderm of the ultimobranchial bodies. So this may be your MCQ in the paper as well, because about its origin the researchers have changed the idea that previously it was thought that these parafollicular cells are originating from the neural crest cell, and now they have changed that they are arising from the ultimobranchial bodies. So by seventh week of the gestation, the thyroid has reached its final destination. So here you can see this is the thyroid gland, and by 7 week it has reached its final destination. Normally this black arrow show the course of the thyroglossal duct, which later on degenerate, and in the area of the tongue we can find the origin which is marked by the foramen cecum. So normally thyroglossal duct degenerate by 10th week of development with only foramen cecum to indicate its former existence. Another important point over here is that this pyramidal lobe, which arises from the isthmus in 50% of the cases, and but also can originate from either lobe, more often from the left as well. So pyramidal lobe may be attached to the hyoid bone by the fibrous band or by the smooth muscle or levator of the thyroid gland. So this pyramidal lobe originating from the isthmus, which is considered as also the distal end of the thyroglossal duct. So what about histogenesis of the thyroid gland? Thyroid primordium consists of solid mass of endodermal cell. So you must know whenever there is an MCQ about the development of the thyroid gland, so you should know that this thyroid gland is endodermal in origin. These endodermal cells aggregation later break up into network of the cords. These cords then divide into cellular groups, and when the lumen is acquired by these groups, the cells are arranged in a single layer around the lumen. By the 11th week, the colloid begins to appear in these structures, and then the thyroid follicles are being formed. So iodine concentration and synthesis of thyroid hormone then can be demonstrated in the embryo as well. So this is again another MCQ, that by 11th week the thyroid follicles are forming, and it is in endodermal origin. So two types of cells, as you all know, they are present in the thyroid. These are the follicular cells and the parafollicular cells. So follicular cells, they arises from endoderm, and parafollicular cells are C cell. They are arising from the ultimobranchial bodies. What about the congenital anomalies of the thyroid gland? So as you know, this originating as a thyroid primordium and going downward and later on connected to initially to its area in the form of the thyroglossal duct. So this thyroglossal duct usually disappear and atrophies, but if it persists or some of its part persists, it can lead to either the cyst or the sinuses. So here you can see the thyroglossal cyst which is present in different part. So the remnant of it may persist in the form of the cyst, either in the tongue or in the anterior part of the neck. So here you can see in the area of the tongue or in the anterior part of the neck. Thyroglossal duct and cyst lies just inferior to the hyoid bone. Its usual location is just inferior to the hyoid bone. Most thyroglossal cysts are observed by the age of the 5 years, and then swelling produced is usually developed as a painless, progressively enlarging movable mass which are present. Then this cyst may contain some thyroid tissue as well. So this here you can see in this area the location, most common location of the thyroglossal cyst which are present. And if these cysts are connected to the outer side by an connection, so these are then called either fistula or sinus, where they will be having their opening as well. Another diagram to show you the thyroid fistula. So here you can see this is the internal opening of the fistula, and here is the external opening of the fistula. So this by definition, fistula is the tract which connects the two epithelial surfaces with each other. So one epithelial surface is that of thyroid area and other is the exterior. So this is the fistula, the thyroglossal fistula which is forming over here. Following infection of the cyst, perforation of the skin occurs, forming thyroglossal duct sinus. It usually opens in median plane of the neck, anterior to the laryngeal cartilages. So again, important point to differentiate between sinus and fistula is that sinus will be only connected to the outer side, to one side, while the thyroglossal fistula will be connected on both the sides. So there will be a free connection. Now we are moving to the development of the parathyroid glands, and first we will see the development of the third pharyngeal pouch over here. So epithelium of the elongate ventral part of third ventral pharyngeal pouch proliferate and their cavities obliterate. Here you can see the first pharyngeal pouch, the second pharyngeal pouch, and then the third pharyngeal pouch which are developing. So from third pharyngeal pouch the inferior parathyroid is forming, and this inferior parathyroid move along with the developing thymus. So as the thymus and the inferior parathyroid both develop from the third pharyngeal pouch, so as the thymus descends, it takes the inferior parathyroid along with it. So inferior parathyroid, although developed from the third pouch, it comes to lie the inferior to the superior parathyroid, and superior parathyroid, it is originating from the fourth pharyngeal pouch. So the primordia of the thymus and parathyroid gland lose their connection with pharynx and migrate into neck. So these arrows show their site of the pathway of the descent. Later the parathyroid gland separate from thymus and lie on dorsal surface of the thyroid gland. So this is the inferior one, and here you can see the two superior parathyroid as well. But so derivatives of the fourth pharyngeal pouch fourth pouch also expands in dorsal, bulbar and elongate ventral part. Its connection with pharynx is reduced to narrow duct, then soon degenerates, and by 6 weeks each dorsal part then develop into superior parathyroid gland. So here you can see this blue color arrow. This shows the dorsal part of the fourth pharyngeal pouch which is forming the superior parathyroid, and it comes to lie on dorsal aspect of the thyroid gland. So the parathyroid gland derived from third pouch descend with the thymus and are carried to a more inferior position than the parathyroid which is developing from the fourth pouch. This explains why parathyroid gland derived from third pair of pouches are located inferior to those from the fourth pouches. So about histogenesis of parathyroid gland, epithelium of the dorsal part of third and fourth pouch proliferate during the 5th week, form small nodule on dorsal aspect of the each pouch. Vascular mesenchyme soon grows into nodules, forming the capillary network, and chief cells differentiate during the embryonic period and regulate the fetal calcium metabolism. The oxyphil cell differentiate 5 to 7 years after birth. So these 2 points again your MCQ as well, that chief cells develop in embryonic period while oxyphil at 5 to 7 years after birth, and they proliferate from 3rd and 4th pouch during the 5th week of development. Elongated ventral pouch of the each fourth develop into ultimobranchial bodies, and these ultimobranchial bodies are then source of the parafollicular or the C cells of the thyroid gland. What about the development of the adrenal gland? So adrenal gland consists of the adrenal cortex and medulla, as you all people know, and it has a distinct albeit interdependent functional properties. So adrenal cortex contains zona glomerulosa secreting mineralocorticoids, zona fasciculata secreting the glucocorticoids, and zona reticularis which are secreting the androgenic hormones. So what about development of the adrenal gland? So adrenal gland is composed of two embryologically distinct tissues, the cortex and the medulla, arising from the mesoderm of urogenital ridge and ectodermal neural chromaffin cell. So it has dual origin again, just like the pituitary gland, the mesoderm giving rise to cortex and ectodermal neural chromaffin cells forming the medulla. So here in this diagram you can see that this pink color area, these are the gonadal ridges over here, and this blue color is the developing suprarenal cortex, and light pink color over here, these are the cells which are forming the adrenal medulla, and these are ectodermal neural crest cell. So here also you can see in blue color the cortex of the suprarenal gland, which is developing from the mesoderm of the urogenital ridge, and in light pink color you can see the medulla, which is forming the chromaffin cell. So an isolated clump of the cells appear within urogenital ridge known as the adrenal gonadal primordium at 28 to 30 days post conception. At 7 to 8 weeks of gestation, the adrenal cortex consists of large inner zone, the fetal zone, and small outer zone, the definitive zone. At the end of the 9th week of the gestation, adrenals become fully encapsulated. After birth, shrinkage of the fetal zone due to increased apoptotic activity occurs, leading to decrease of the weight of the adrenal gland by 50%, and this is again your another MCQ as well. So this is the reason why the weight of the adrenal gland decreases after birth. This is because of the apoptosis of the fetal zone, not that of the definitive zone. Definitive zone remains as such. Fetal zone, which was initially very larger in size, it shows apoptosis and shrinkage, as a result of which there is 50% decrease in weight of the adrenal gland. So this is all about the development of the adrenal gland. Here are the references for today's class, that is Langman's Medical Embryology, the Developing Human by Keith Elmore, and Gray's Anatomy by the Susan Standrings, the 40th edition. Thank you very much for your attention, and Allah Hafiz till the next class.